All right, my challenge today is to introduce you to a compound microscope. As it happens, Denver Christian provides really actually very nice student microscopes. It's important that you understand how they function so that you know both how to take care of them and also how to use them effectively. So I'm going to talk first about just kind of the physical equipment that's necessary, not to help you see an image, but sort of necessary to make the thing kind of work. And you're going to want to take notice of the names of the parts and the functions of the parts as we go along. So first of all, obviously the microscope has to sit on something, and that thing that it sits on is down here, and it's called the base. So there's the base of the microscope. The base contains some form of illumination, so in this case it contains a light. There's a light switch the light it goes on, obviously, if the microscope is connected to a power source. The rest of the microscope is held in place by this structure right here, which is technically called the arm. And when you carry a microscope, you carry the microscope with two hands, one hand holding the arm and the other hand supporting the base. All right? Now, how does the microscope work? What happens when you look at a specimen under the microscope? Well, first of all, we will talk later about how to prepare the specimen for viewing. But first of all, you probably already know where you put the specimen. You put the specimen on this platform, which is called the stage. And if you look carefully at the stage, you'll see that there are little metal clips here. Those are called stage clips. They hold the slide in place. And what you want to do is you want to position the slide so that the specimen, if you can see it, or the area that you imagine the specimen to be in, if you can't see the specimen, is centered directly over the hole in the stage. And you probably can't see it on the video, but when you actually work with the microscope, that hole will be easily seen. It'll be illuminated from below by the lamp. So let's imagine that you have a slide on the stage, your specimen is centered over the hole in the stage. What basically do you see, and how is it that the microscope produces the image that you see? Well, the light comes up from the base and it goes into the bottom of the stage. And again, it's going to be a little tricky to see in the video, but underneath the stage there's a round disc, and that round disc is called a condenser. And what the condenser does is it focuses the light of the beam of light from the lamp up through that hole in the stage. All right? You can't really see the condenser clearly. What you can see is an additional piece of equipment called the diaphragm. This happens to be the diaphragm. You can see me turning. All right? It's actually the disc portion of the condenser with a series of 5 holes. And the 5 holes are of different sizes, so as you change the position of the disc diaphragm, more or less light gets shined up through the hole in the stage. So basically there are 2 parts here. One is a condenser. It focuses the beam of light on the specimen, and the other is the diaphragm. And the diaphragm controls the amount of light that actually gets to the specimen. Typically, when you use a microscope, you want to use the diaphragm at the brightest possible size, so that would be position number 5. We'll talk about that when we talk about troubleshooting a little bit. All right, so now the light comes up through the specimen. It's obviously going to pass through the air at this point into one of these lenses. And if you look closely, you'll see that what I have here is I have a revolving structure. This is called a revolving nose piece, and hanging down from the revolving nose piece are 3 objective lenses. The 3 objective lenses are different lengths. I have in position right now the shortest of the 3. That's called the scanning lens. The next longer lens is called the low power lens, and the longest lens is called the high power lens. Be gentle when you turn the revolving nose piece. You'll be able to feel that the lenses, the objective lenses, click into place. You want to make sure it clicks firmly into place. If it's a little bit off, you won't be able to see anything. When we use the microscope, we always start with the lowest power objective in place, and that's always the shortest objective. The scanning objective on these microscopes magnifies the image 4 times. So the light passes through the sample, goes into the objective lens, and the objective lens magnifies the image of that specimen 4 times. The light passes then upward through this portion of the microscope right here, which is called the body tube. There's a set of mirrors in the body tube that can change the path of the light slightly so it comes up through this portion of the microscope and ultimately out the ocular lens. This is called an ocular or eyepiece. And inside the ocular is another lens. And this lens will magnify the image either 10 times or 15 times, depending on what kind of microscope you have. Here at Denver Christian, some of the oculars are 10x, some of them are 15x. This one happens to be a 15x ocular. So let's talk for a moment about total magnification. If the scanning lens has magnified the image 4 times, and then the ocular has magnified the image another 15 times, our total magnification is 4 times 15. So our total magnification is 60x. So the scanning lens is called a scanning lens because it doesn't magnify very much. Its purpose is to allow you to scan a wide surface of your slide and hopefully locate your specimen. Once you've located a specimen, all right, you need to obviously adjust magnification. You adjust magnification by turning the revolving nose piece, snapping another objective lens in place. The low power objective magnifies the image 10 times, and then the high power objective magnifies the image another 43 times. So if you do the math, you can see that you have a pretty wide range of magnification possible. Now, I've obviously skipped past one very important aspect of microscope function, and that is the whole issue of focusing. All right, if you look closely as I turn this knob, you'll see that the stage moves up and down. The movement of the stage up and down is how we bring about the focusing of the image. Now, this may seem a little bit counterintuitive, all right, but the first bit of really good advice I can give you on looking for something under a microscope is always start with the lowest power available and always start with the stage in the highest possible position. All right, now you're going to find that these microscopes are good student microscopes. They have a stop, which means that you're not going to be able to jam the slide into the bottom of the objective lens. That's a good thing. Objective lenses are expensive. So rack the stage all the way up to its highest point until it stops using this big knob. This big knob is called the coarse focusing knob. And then, looking through the ocular, begin to focus down and focus very, very slowly down. As you focus down, things should come into focus and then go back out of focus. And once you find that place where the specimen's coming in and then goes out of focus as you move it, you obviously get it, then you back it back up, back into focus, and then you use this smaller of the two knobs, which is the fine focus knob, and you make a final adjustment. One of the things that is really nice about these microscopes is that they are approximately parfocal. And what that means is that once you have the specimen in focus under your scanning lens, you should be able to switch to your low power lens without changing the position of the stage, and the specimen should be just about in focus. All you should have to do is adjust with the fine focus knob just a little bit. And then, even better news, the microscope should just about be in focus for the high power objective. So again, even though it seems impossible, you should be able to turn, and I'm not going to try it here because I don't have a specimen that's in focus, but if I have a specimen that's in focus, I ought to then be able to turn my high power objective in place. It will it'll look like it's going to touch that slide, but it shouldn't, it probably won't, and again, your image should be just about in focus. With a little adjustment of the fine focus knob, you should be there. Good luck.